Hello everyone, this is Toxai brought to you by Waitira. Most people call me Carol Ndegwa. It is quite a cold August here in Eldoret. I'm really not complaining. I really do love the cold weather. It's another week, and so we do have a new episode out. Today I'll be discussing about plant tissue culture. I thought it's important or relevant for my audience to understand or know what plant tissue culture is, since in the last two episodes I have mentioned these techniques or this technology. In our second episode, I mentioned about plant tissue culture being used in conserving biodiversity. I mentioned about the synthetic seed technology being used to conserve species that are threatened by climate change or natural disasters or even human intervention. In the third episode, I mentioned about plant tissue culture being used in genetic engineering to ensure that certain superior traits are introduced into another crop. And therefore, with me mentioning plant tissue culture, I thought it's really important for you as my audience to understand what is this that I keep, mentioning. But not only that, my audience is able to understand either is there a farmer here, they're able to understand how plant tissue culture can contribute to the agricultural productivity. You as a consumer, you're able to understand the food that you're eating or consuming from the market and the high nutritional value that you're getting maybe from a sweet potato or even from, maize, and how plant tissue culture is playing a role to ensure that you are not getting malnutrition. But even for any biotech student who is here, they're able to utilize plant tissue culture techniques to be able in their research work or in their study work, because tissue culture is broad. It extends to either plants or animals. As such, in animals you will look at embryo fertilization, especially in cows. And therefore, I'd like us to dive into plant tissue culture, and this is a production of plants on specially formulated nutrient media under sterile conditions. This nutrient media normally mimics soil that you would rather find in your shamba. It contains the basic components that are required for plant growth. That means it has the water, the macronutrients, the vitamins, the salts, and even the micronutrients that are required for plant growth. And plant tissue culture, or otherwise PTC, it has become feasible on the basis of two principles. One of these principles is totipotency, which is the ability of any part of a plant to grow into a whole plant. This means that if I just had a root or a flower or a seed or even a leaf, I will just use that part of the plant to grow into a whole plant. And also the next principle is plasticity, which is the ability of a crop to grow in any environment, even though it is not its habitat, but as long as the growth conditions are provided. And it is these two principles that have enabled scientists to manipulate plants, and especially in plant breeding programs. And so any individual would ask me, Carol, why are you trying so hard to do this plant tissue culture thingy? And this is because plant tissue culture has advantages compared to the conventional methods of propagation, such as the use of seed, the use of splits, or even the use of cuttings. And one of these advantages is the production of disease-free plants. So I have this popo plant that is really high yielding. It gives me really big juicy purple fruits, but it is infected by systemic disease. Systemic meaning I am not able to control it using maybe a fungicide. And so I will use a meristem culture to produce disease-free plants. I would like you to envision when you have a seed in the ground, and then the seed, after 2 weeks, the seed has already emerged. About a month, it has grown to 30 centimeters. About 2 months, it's already about 90 centimeters. There's this part of the plant that is always growing, and this part of the plant is called the meristem tissue. The use of the meristem tissue, because it's normally very actively dividing, it has cells that are very actively dividing, and therefore it does not allow disease to get to it. And so scientists are able to use that part of the plant to regenerate a whole plant. Another advantage of plant tissue culture is mass propagation, where this technique enables scientists to produce large quantities of crops within just a small space. When you envision, when you have a stem and it has several nodes, and every node you can be able to regenerate it into a whole plant using nodal cultures. Another advantage of plant tissue culture is the propagation of recalcitrant crops. Plant breeders have been able to utilize hybridization techniques in crop improvement. Hybridization is, in simple terms, is when you have a male plant and a female plant, you cross the two to get an offspring. Normally, this offspring is genetically different to the male and to the female plant. And sometimes after fertilization, the embryo is weak and it is not able to grow into a whole plant. Also, another scenario is sometimes during pollination, the pollen is not fertile enough to be able to fertilize the ovule, and therefore fertilization doesn't even take place. And therefore, tissue culture comes in handy to enable scientists to be able to cross these two plants that are not compatible, or once after fertilization has taken place and an embryo has developed but this embryo is weak, what is done is an embryo rescue, so you are able to use that embryo to regenerate a whole plant. Another advantage of plant tissue culture is during plant transformation processes, and that is basically genetic engineering. And the products of genetic engineering normally contain two DNAs, that it has its own DNA and it has a foreign DNA that has been introduced. And normally the DNA that has been introduced is to enable this plant to be tolerant to either biotic or biotic stresses, or maybe to be high yielding. And for this foreign DNA to be introduced into the plant, tissue culture techniques have to come into play. And they are normally used especially during co-culturing to enable, co-culturing is when you culture the agrobacterium, which is a vector, and the plant that you want to transform. And during that co-culture is when the trait is introduced into the plant, and also during trait identification, because you want to see whether really the trait you are trying to introduce actually got into the plant. Another advantage of this technique is biodiversity conservation, where scientists are able to use the synthetic seed technology to conserve species that are vulnerable to erosion. So these synthetic seeds can stay to periods of months or even years, even 10 years. And, once breeders or individuals would want to utilize this as species, they are able to go to gene banks and, access them and use them. However, plant tissue culture comes with its own disadvantages. With every pro, there is a con. One of these disadvantages is sometimes these procedures can take really, really long time. Remember, we are regenerating, for example, when we are trying to do disease-free plants, we are regenerating a plant from a meristem tissue. And sometimes it can take periods of even 7 months to be able to regenerate a whole plant. So sometimes it can take a really long time before you are able to, get your whole plant. But again, when you look at the advantage, you'll rather wait, right? Another disadvantage is that plant tissue culture requires skilled labor, and sometimes this skilled labor can also be, expensive. And because you need the knowledge of doing laboratory operations, accessing a meristem tissue, so you require people who are knowledgeable about these things. Another disadvantage is plant tissue culture is really expensive. All these reagents that are used in plant tissue culture are really not cost effective, and therefore you will find that even a farmer is not able to do plant tissue culture in their own home, because you require even equipment such as the laminar flow, which is really expensive. Reagents, I mentioned about the nutrient media. The one we use at the lab is called the MS media, and it is really expensive. And so it wouldn't even be cost effective to a farmer because it wouldn't, return the investment. And therefore, this plant tissue culture can only be done in a specialized laboratory with certain standards. And, it can only be done by organizations who are able to fund these approaches. But however, I normally look at the disadvantages of plant tissue culture as something that is worth, I mean, when you look at it, when you look on the brighter side, there are advantages. I mean, why would I struggle with a purple plant that is that has disease when I could just be a little bit patient and get a whole plant that is high yielding and is disease free? I mean, and also when it comes to skilled labor, we have very many biotech students who are getting into biotechnology, and so we already have the skilled labor. And also when it comes to the expensive approach, plant tissue culture can be really expensive. It is really expensive. And so that one, I really do not see how, a farmer can be able to do that on their own. But we have all these stakeholders who are working together, and to even lift the weight on other stakeholders. Like you will find Calro or even JQuat producing numerous uh, banana plantlets and distributing them to farmers. So with all these stakeholders working together, you find that when it comes to the cost effectiveness, they are able to counter it. I mean, I'm just trying to make you see that as much as PTC has its own disadvantages, we can work around them, considering the advantages that it provides. And so I would like us to get a little bit more practical and see how tissue culture techniques are being used in the real world. Ornamental trade is a significant source of foreign exchange in South Asia, and countries like Singapore, Thailand, and also Malaysia are able to improve their economic growth through ornamental trade. And because of the high demand that these countries have, they use tissue culture technology to be able to mass propagate these plants. Remember, I also mentioned about ornamental trade or the ornamental industry, the aspect of uniformity being very key, and therefore tissue culture comes in with clonal propagation to be able to meet the market demands and need and also preference. A country like Thailand is able to produce about 50 million plantlets annually, and I mean, all props to plant tissue culture. In Kenya, we are able to produce banana plantlets and distribute them to farmers. In about 20 years ago, there was a problem with banana production in Kenya, and this was majorly because of soil degradation. There were emerging pests and diseases, and also the poor agronomic practices by the farmers. And organizations such as Calro, there's also JQuat, they came in and did a lot of production of banana plantlets, distributing them to farmers. Not only are these plantlets being produced to farmers in very high quantities, one, they meet the demand, but they are also very high yielding. I was reading a bunch could weigh about 30 to 45 kg as compared to what the farmers would plant quite a while ago, which was they would harvest, I mean, quite a while ago, which was about 10 to 15 kg per bunch. And you can see how tissue culture is really improving the economic livelihoods of these small scale farmers. In West Africa, tissue culture came in handy in plant breeding programs. Scientists were able to use embryo culture and also anther culture. Anther culture is where you use anthers as your starting material and you regenerate it into a whole plant. And they were able to use these two techniques, the embryo and the anther culture, to cross two incompatible rice varieties. And the final variety that they produced was not only high yielding, but it also had traits that enabled it to tolerate the environmental stresses. Those are just some of the examples in which tissue culture is being used in the real world. There's also the use of tissue culture in cassava propagation, in improvement of pyrethrum, and you can see how tissue culture comes in handy, especially in agricultural development, not only in Kenya, not only in Africa, but globally. As always, guys, thank you for being here. Like, share, and subscribe, and see you on the next one. Churros.